Stem cells are undifferentiated biological cells in multicellular organisms that can differentiate into specialized cells and can divide to produce more stem cells. A stem cell is essentially a blank cell that is capable of becoming another more differentiated cell type in the body, such as skin cell, a muscle cell or a nerve cell. They are microscopic in size and can be used to replace or even heal damaged tissues and cells in the body. In a developing embryo, stem cells can differentiate into all the specialized cells like ectoderm, endoderm and mesoderm and maintain the normal turnover of regenerative organs such as blood, skin or intestinal tissues. In adult organisms, stem cells and progenitor cells act as a repair system for the body, replenishing adult tissues. The history of research on adult stem cells began more than 60 years ago. Scientists first studied the potential of stem cells in mouse embryos over 2 decades ago. It was only in 1998 that researchers first extracted the first human embryonic stem cells that were able to be kept alive in the laboratory. The embryonic stem cells can be taken from umbilical cord blood just after birth. The autologous cells are obtained from one's own body. The embryonic cell lines and autologous embryonic stem cells generated through somatic cell nuclear transfer or dedifferentiation have also been proposed as promising candidates for future therapies. There are 3 known sources of autologous adult stem cells in humans: bone marrow, from which stem cells are extracted by drilling into bone of femur or iliac crest; adipose tissue, from which stem cells are extracted by liposuction; blood, from which stem cells are extracted through apheresis and passed through a machine that extracts the stem cells and returns other portions of the blood to the donor. Stem cells differ from other kinds of cells in the body. The classical definition of a stem cell requires that it possesses 3 properties: self-renewal. The stem cells may replicate or proliferate many times, that is, it has the ability to go through numerous cycles of cell division for long periods while maintaining the undifferentiated state. Potency. The stem cells should be potent to exhibit totipotent or pluripotent, that is, the capacity to differentiate into specialized cell types. Unspecialized. One of the fundamental properties of a stem cell is that it does not have any tissue specific structures that allow it to perform specialized functions. These unspecialized stem cells can give rise to many different specialized cells. The process of differentiation is controlled by internal and external signals. The interaction of signals during differentiation causes the cell's DNA to acquire epigenetic marks that restrict DNA expression in the cell and can be passed on through cell division. The function of stem cell is regulated by a feedback mechanism. In mammals, there are 2 broad types of stem cells: embryonic stem cell, which are isolated from the inner cell mass of blastocysts, and adult stem cells, which are found in various tissues. The embryonic stem cells are derived from a 4 to 5 day old human embryos in the blastocyst phase of development. The embryos are usually extras that have been created in vitro fertilization clinics, where several eggs are fertilized in a test tube, but only one is implanted into a woman. Or they may be donated for research purposes with informed consent of the donors. The zygote, after 4 to 6 days of fertilization, becomes a mass of cells called a blastocyst. The blastocyst consists of an inner cell mass and an outer cell mass. The outer cell mass becomes part of the placenta, and the inner cell mass differentiate to become all the structures of an adult organism. This mass is the source of embryonic stem cells called totipotent cells. When extracting embryonic stem cells, the blastocyst stage signals when to isolate stem cells by placing the inner cell mass of the blastocyst into a culture dish containing a nutrient-rich broth. In the absence of stimulation to differentiate, they begin to divide and replicate while maintaining their ability to become any cell type in the human body. Eventually, these undifferentiated cells can be stimulated to create specialized cells at various points during the process of generating embryonic stem cell lines. Scientists perform characterization to see the viability of these cells. They exist through the body after embryonic development and are found in tissue and organs including brain, bone marrow, peripheral blood, blood vessels, skeletal muscle, skin, teeth, heart, gut, liver, ovarian epithelium and testes. They are thought to reside in a specific area of each tissue called a stem cell niche. They can differentiate into cells of same lineage. The adult stem cell can renew itself indefinitely and can differentiate to yield some or all of the major specialized cell types of the tissue or organ. The primary roles of adult stem cells in a living organism are to maintain and repair the tissue in which they are found. Stem cells may remain quiescent for long periods of time until they are activated by a normal need for more cells to maintain tissues or by disease or tissue injury. Scientists have found adult stem cells in many more tissues than expected, like in brain and heart. This finding has led researchers and clinicians to consider use of adult stem cells for transplants. If the differentiation of adult stem cells can be controlled in the laboratory, these cells may become the basis of transplantation-based therapies. Now, let us see the similarities and differences between human embryonic and adult stem cells. Both have advantage and disadvantage regarding potential use of cell-based regenerative therapies. One major difference between adult and embryonic stem cells is their different abilities in the number and type of differentiated cell types they can become. Embryonic cells can become all cell types of the body because they are pluripotent, while adult stem cells are thought to be limiting to differentiating into different cell types of their tissue of origin. The embryonic cells can be grown relatively easily in culture, while adult stem cells are rare in mature tissues, so isolating these tissues from an adult tissue is challenging. The adult stem cells and tissues derived from them are less likely to initiate rejection after transplantation by the immune system. This is because a patient's own cell could be expanded in culture, coaxed into assuming a specific cell type and then reintroduced into the patient. However, embryonic stem cells may initiate rejection. Differentiation. Stem cells can differentiate into hematopoietic stem cells, which give rise to all types of blood cells: RBCs, lymphocytes, natural killer cells, granulocytes, monocytes and macrophages. Mesenchymal stem cells that have been reported to be present in many tissues. Those from bone marrow stromal stem cells, skeletal stem cells give rise to a variety of cell types like bone cells and cartilage cells, fat cells and stromal cells that support blood formation. Neural stem cells in the brain give rise to nerve cells and non-neuronal cells like astrocytes and oligodendrocytes. Epithelial stem cells in the lining of the digestive tract, it occurs in the deep crypts, giving rise to several cell types like absorptive cells, goblet cells and enteroendocrine cells. Skin stem cells, they occur in the basal layer of the epidermis and at the base of hair follicles. The epidermal stem cells give rise to keratinocytes which form a protective layer. The follicular stem cells give rise to both the hair follicle and epidermis. Transdifferentiation. Many experiments have reported that certain adult stem cell can differentiate into other cell types outside the cell's predicted lineage. For example, the brain stem cells differentiate into blood cells or blood forming cells, differentiate into cardiac muscle cells, etc. by the process called transdifferentiation. Scientists demonstrated that this may be because of well-controlled process of genetic modification. This strategy may help to reprogram available cells into other cell types that have been lost or damaged due to disease. For example, if the pancreatic beta cells that produces insulin are lost or damaged in diabetes, it could possibly be created by reprogramming other pancreatic cells that can secrete insulin. Adult somatic cells can be reprogrammed to become embryonic stem cells and are known as induced pluripotent stem cells or iPSCs. Thus, a source of cells can be generated that are specific to the donor, thereby increasing the chance of compatibility during tissue regeneration. Growing cells in the laboratory is known as cell culture. Stem cells are either extracted from adult tissue or from a dividing zygote in a culture dish. Once extracted, scientists place the cells in a controlled culture that prohibits them from further specializing or differentiating further, but allows them to divide and replicate. The process of growing large numbers of embryonic stem cells has been easier than growing large numbers of adult stem cells, but progress is being made for both cell types. Tissue collection and transportation. The first and most important element in the collection of tissue is the cooperation and collaboration of the clinical staff, such as the surgical, one of the nursing staff or pathologist. The tissue should be collected in a double wrapped sealed polythene bag or container and an outer padded envelope provided with the name, address and phone number of the recipient at the laboratory. The label should also state clearly, "Do not freeze." Refrigeration during transport is not usually necessary unless the delivery takes more than an hour or two, which can be done with refrigeration packs. The tissue will be used to corroborate the origin of any cell line that is derived from the sample by DNA profiling. Biosafety and ethics. All procedures involved in the collection of human material for cell culture must be passed by the relevant hospital ethics committee. A form will be required for the patient to sign authorizing research use of the tissue and preferably disclaiming any ownership of any materials derived from the tissue. All human material should be regarded as potentially infected and treated with caution. Each laboratory will have its own biosafety regulations that should be adhered to. Record keeping. When the samples arrive at the laboratory, it should be entered into a record system and assigned a number. The record should contain the details of the donor, tissue site, collection medium, time in transit, treatment on arrival, primary desegregation and culture details, etc. The embryonic stem cells, called HESC, are generated by transferring cells from a pre-implantation stage embryo into a plastic laboratory culture dish coated with mouse embryonic skin cells, specially treated not to divide. This coating layer of cells is called a feeder cell or feeder layer. The culture dish contains a nutrient broth known as culture medium. The cells divide and spread over the surface of the dish. The mouse cells in the bottom of the culture dish provide the cells a sticky surface to which they can attach. Also, the feeder cells release nutrients into the culture medium. Researchers have now devised ways to grow embryonic stem cells without mouse feeder cells. This is a significant scientific advance because of the risk involved, where viruses or other macromolecules in the mouse cells get transmitted to the human cells. If the plated cells survive, divide and multiply enough to crowd the dish, they are removed gently and plated into several fresh culture dishes. The process of replating or subculturing the cells is repeated many times for many months. Each cycle of subculturing the cell is referred to as passage. Once the cell line is established, the original cells yield millions of embryonic stem cells. At any stage in the process, batches of cells can be frozen and shipped to other laboratories for further culture and experimentation. Some examples of potential treatments include regenerating bone using cells derived from bone marrow stroma, developing insulin producing cells for type 1 diabetes, and repairing damaged heart muscles following a heart attack with cardiac muscle cells. Once stem cells have been allowed to divide and propagate in a controlled culture, the collection of healthy, dividing and undifferentiated cells is called a stem cell line. Once under control, the stem cells can be stimulated to specialize as directed by a researcher, a process known as directed differentiation. Embryonic stem cells are pluripotent and are able to differentiate into more cell types than adult stem cells. Stem cells are categorized by their potential to differentiate into other types of cells. Embryonic stem cells are the most potent since they must become every type of cell in the body. To confirm experimentally that a putative adult stem cell is indeed a stem cell, scientists tend to show either that the cell can give rise to these genetically identical cells in culture and or that a purified population of these candidate stem cells can repopulate or reform the tissue after transplant into an animal. Laboratories that grow human embryonic stem cells use several kinds of tests, including growing and subculturing the stem cells for many months to ensure that the cells are capable of long-term growth and self-renewal. The cells must be healthy and undifferentiated. Using transcription factors like OCT4 and NANOG, which are used to maintain the stem cells in an undifferentiated state capable of self-renewal. These help turn genes on and off at the right time to ensure cell differentiation and embryonic development. Use of specific techniques to determine the presence of particular cell surface markers typically produced by undifferentiated cells. Testing whether the human embryonic stem cells are pluripotent by number 1, allowing the cells to differentiate spontaneously in cell culture. Number 2, manipulating the cells so they will differentiate to form cells characteristic of the 3 germ layers. Number 3, injecting the cells into a mouse with a suppressed immune system to test for the formation of a benign tumor called a teratoma. Although complete agreement among scientists as to how to identify stem cells are not prevalent, yet most tests are based on making sure that stem cells are undifferentiated and capable of self-renewal. Preservation of cells for a long time are done by freezing secure stocks to be maintained without aging and protect them from problems of contamination, incubator failure or medium and serum crisis. Ideally, 10 to the power 6 to 10 to the power 7 cells should be frozen in 10 ampoules in glycerol or dimethyl sulfoxide at 5 to 10%. Smaller stocks can also be used as per requirement and availability. The normal procedure is to freeze a token stock from the surplus cells and expand remaining cultures to confirm the identity of the cells and absence of contamination. Next, a seed stock of 10 to 20 ampoules are frozen down. 1 ampoule thawed from this stock can then be used to generate a using stock. Cross-contamination. This is less of a problem with short-term cultures, but the risk remains that if there are other cell lines in use in the laboratory, they can cross-contaminate even a primary culture, or misidentification can arise during subculture or recovery from the freezer. Hence, precautions must be taken to avoid cross-contamination, namely by not handling more than one cell line at a time, not having culture vessels and medium bottles for more than one cell line open at one time, by avoiding the use of same pipette or other device for different cell lines, by not sharing media or other reagents among different cell lines or with other people, by ensuring disinfecting with 70% alcohol during any spillage immediately, by retaining a tissue or blood sample from each donor and confirm the identity of each cell line by DNA profiling during freezing seed stock or before the cell lines is used for experimental work or transplantation, by keeping photographs of each cell line at low and high densities and consult this regularly when examining cells during maintenance. To avoid microbial contamination, antibiotics are often used during collection, transportation and dissection of biopsy samples because of the intrinsic contamination risk of these operations. If the culture grows well, then antibiotics can be removed from the bulk of the stocks at first subculture, retaining one culture in antibiotics as a precaution if necessary. Antibiotics can lead to lax aseptic technique. They can inhibit some eukaryotic cellular processes and also hide the presence of a microbial contamination. If a culture is contaminated, the culture should be discarded before it can spread the contamination to other cultures. Most bacterial, fungal and yeast infections are readily detected by regular, careful examination with the naked eye, for example, by a change in the color of the culture medium and on the microscope. There are many ways in which human stem cells can be used in research and the clinic. A more complete understanding of the genetic and molecular controls of these processes may yield information about how such diseases arise and suggest new strategies for therapy. Stem cell therapy uses stem cells to treat or prevent a disease or condition. Bone marrow transplant is a crude form of stem cell therapy that has been widely used clinically for many years without controversy. Research is underway to develop various sources for stem cells and apply stem cell treatments and research. Stem cells help in basic research such as clarification of complex events that occur during human development and understanding molecular basis of cancer. Molecular mechanisms for gene control signals in gene expression and differentiation. The undifferentiated stem cells eventually differentiate partly because of a particular gene is turned on or off. Stem cell researchers may help to clarify the role that genes play in determining what genetic traits or mutations we receive. Understanding the theory of cancer. Stem cells help in cell-based therapies such as regenerative therapy to treat Parkinson's, Alzheimer's, spinal cord injury, stroke, severe burns, heart disease, diabetes, osteoarthritis, wound healing and rheumatoid arthritis. The learning disability due to congenital disorder, anti-cancer treatments, repair hearing and restore vision and repair damage to the cornea and amyotrophic lateral sclerosis or ALS. Organ and tissue regeneration is probably the most important possible application of stem cell research. Stem cells could potentially be used to grow a particular type of tissue or organ. Stem cells that lie just beneath the skin, for example, have been used to engineer new skin tissue that can be grafted onto burn victims. Healthy heart cells developed in a laboratory may be transplanted into patients with chronic heart disease, repopulating the heart with healthy tissue. Similarly, type 1 diabetes can be cured by directing the differentiation of human embryonic stem cells in cell culture to form insulin producing cells that can be eventually used in transplantation therapy. Adult hemopoietic stem cells found in bone marrow and blood have been used for years to treat blood diseases such as leukemia, sickle cell anemia and other immunodeficiencies. These cells are capable of producing all blood cell types such as RBCs that carry oxygen to white blood cells that fight disease. The cells are extracted from bone marrow or umbilical cord and placenta. Stem cells help in biotechnology such as in drug discovery and development by providing specific cell types to test new drugs. In screening of potential drugs, human stem cells are currently being used to test new drugs. New medications are tested for safety on differentiated cells generated from human pluripotent cell lines. Cancer cell lines are already being used to screen potential anti-tumor drugs. The availability of pluripotent stem cells would allow drug testing in a wider range of cell types to reduce animal testing. Harvesting of embryonic stem cells in order to avoid the problem of immune rejection, scientists are experimenting with different research strategies to generate tissues that will not be rejected. One of the disadvantages of stem cell treatments is that it may require immunosuppressant because the patient may receive radiation before the transplant or because the patient's immune system may target the stem cells. It is therefore preferable to use stem cells from the same patient who is being treated. Pluripotency in certain stem cells could also make it difficult to obtain a specific cell type. It is also difficult to obtain the exact cell type needed because not all cells in a population differentiate uniformly. Undifferentiated cells can create tissues other than desired types. Some stem cells form tumors after transplantation in embryonic stem cells. Fetal proper stem cells induced pluripotent stem cells. Fetal proper stem cells form tumors despite multipotency. Scientists and researchers are interested in stem cell cultures for several reasons. Stem cells extracted can be induced to become any desired cell type. This property makes them powerful enough to regenerate damaged tissues under the right conditions. However, the use of stem cell is still under controversy. Stem cell research helps to identify how undifferentiated stem cells become the differentiated cells and understand about the complex events that occur during human development. A more complete understanding of the genetic and molecular controls of these processes may yield information about how such diseases arise and suggest new strategies for therapy. Stem cell therapy uses stem cells to treat or prevent a disease or condition. To summarize, stem cells offer exciting promise for future therapies, but significant technical hurdles remain that will only be dealt with through years of intensive research. In the coming years, with increasing ability of scientists to isolate and culture embryonic stem cells and grow induced pluripotent stem cells, we can hopefully reach the zenith of treatment of diseases.